Good morning. Welcome to the keynote session of Discovery Forum 7. I'd like to very warmly welcome everyone who made it in person to our small venue, and to also welcome the whole audience that has been joining us throughout the day online. My name is Kate Witkowska. I'm the Strategic Partnerships Director at Genomics England, and my key role here is to work with our pharma partners in enabling them to leverage the value of our data set, but also to go beyond that and to really translate the findings into potential benefits for our patients. So it is my tremendous pleasure today to welcome our keynote speaker for the day, Pierre Farmer, who is the Associate Director of Data Science at Novartis Institute for Biomedical Research. Pierre has extensive, academic background in pharmacology and bioinformatics before he transitioned into industry, first at Merck KGaA and then more recently at Novartis. More importantly for our GEL Novartis relationship, Pierre has been an instrumental point of contact for us, really setting up, growing and nurturing our Novartis Genomics England collaboration from the very beginnings of the Discovery Forum 2018-2019. And over the 3 years of our partnerships working, he's been really a true champion, as you will be able to witness from his talk, bringing together over 80 scientists across Novartis to ideate over 10 live projects in our research environment, and some of these have now potential to be translated further. And so Pierre will talk about all of these stories in his talk, hopefully showing you what value can be leveraged from the Genomics England dataset, but also to stimulate a discussion about how partnership working can help us translate that into even more benefit. So without further ado, Pierre, welcome. It's a shame we can't have you in person, but welcome very much virtually, and over to you.
Thank you. Thank you so much, Kate, for these kind words. And I would like first to express all my gratitude to the organizing committee for the opportunity to present today, and also for this amazing team, which I'm presenting on their behalf. So our journey with Genomics England really started a few years ago, where our interest was really to find a genetic data set in order to identify and prioritize targets. But what quickly emerged is that when genetic data becomes linked to electronic health record, also with the possibility to access biobank samples and also recall patients, this opens up all sorts of opportunities that are really enterprise-wide applicability across the pipeline. And in this presentation, I will present a few vignettes from very early discovery all the way to medical affairs on how we are applying at Novartis genomic medicine resources such as Genomics England. And I will ask you for a little bit of patience on my side on the fact that in some of these vignettes I can be very precise. In others, they will be a little bit more vague because I cannot disclose all items, but I'm sure you will understand. One very interesting aspect also is, in theory, we can really perform all these steps with the same population cohort from target identification to clinical trial. And this is kind of relatively new and really puts the patient at the center of our pipeline. And finally, the combination of omics with real-world data brings new ways to study markets or to study real-world data. And also through understanding of disease heterogeneity can open up new target possibilities, therefore transforming a linear pipeline into a more circular value creation loop, very analogous to Chris' infinity loop concept. If we go to the next slide, the first vignette is around target identification and prioritization. So here I would like to really point out the great work of our colleagues in other pharmas, notably AstraZeneca, GSK, BMS, where AbbVie, who they presented that target indication pairs for which there is genetic evidence supporting the association increases the likelihood of a successful phase 2 by double, increases doubles the likelihood of a successful phase 2 trial. And that attracted a lot of interest within Novartis into look much more closely into genetic data and how we are using a gel platform within that landscape. Is that, well, we very quickly realized that due to the disease fragmentation, this was not the data set that is most ideal to do brand new GWAS analysis, particularly in non-rare disease indication or non-oncology indication. But where the gel resource turned out to be very valuable, it is to have this enrichment in Mendelian-like mutations that are present in different target genes that can also unveil quite information regarding its biology. For example, what is the consequence of having a missense mutation in a binding pocket? And that can also inform the building a modality against this target. But the data is mostly used to do safety type of analysis and also to confirm or to use the data set as a meta-analysis in conjunction with other platforms. But one surprising area that we did not expect where gel was extremely useful is for modality development. And the first example brings the context where I mentioned before that Novartis is not traditionally put a lot of effort in genetics, but it did prioritize work on phenotypic screens. And what we have learned is having very, very relevant biological assays in order to perform these screens really helps the likelihood that a compound will be relevant in a clinical context. And here, accessing iPS cells in order to differentiate them into mini organs or relevant cell types under the disease background proved to be very useful. So with gel, we find the capability of recalling phenotyped and genotyped patients in order to get a blood donation, and from these patients derive iPS cells that can be used in order to do functional validation of a gene pathway, particularly in cases where these patients are carrying loss of function or gain of function variation in the target of interest, and also enables us to understand the genetic consequence of the hyperactivity or hypoactivity of a specific pathway in contrast to other pathways. This is something that turns out to be quite useful. And of course, the same iPS cells can be used in a context of a mid-size modality screen. The next example or vignette is also in the field of modality building, but in a different context, which is here gene therapy. So here I cannot disclose the disease nor the target, but what is really relevant to know here is that a single loss, a copy loss of a gene causes the disease of interest. And of course, we are in a process of providing a cure for this disease through a gene therapy strategy. Now, the challenge in this particular project is that there's a very high homologous gene sequence overlap between preclinical models, for example, mice or non-human primates and human. Therefore, in PK/PD studies or in toxicology studies, it is not possible to distinguish if the protein that is found in the target tissue, if it was expressed using the wild type copy or the host copy, or if it was expressed through the transgene copy. And this discrimination is crucial for a successful PK/PD study. And the solution that the team provided was to find missense mutation that on one hand would allow discrimination of the transgene from the host gene copy, but at the same time would not impact the biological function of that protein. So the team came with a flowchart-like analysis where gel was the data set of interest because it has deep phenotyping of the patients and also has a multi-disease cohort, which was of great interest for us. So the approach was really to exclude patients with a target disease, but using a definition of that disease that is much broader than the intent indication. And of this, we identified 332 potential missense mutation. And through different analysis, we prioritize 2 missense variants that on one hand, we are quite confident that does not impact the function of that protein, but on the other hand, enables the distinction of the transgene copy versus the host copy using MS technology. Now, on the next slide is more a use case around indication search on a program that is completing its discovery process. So there's a Novartis team that built an oral low molecular weight therapy compound for therapies or indications that are caused by mutation, premature termination codon mutation in given genes. So a PTC is a mutation where a stop codon is introduced, and during translation, the protein, the translation stop at that stop codon, generating a dysfunctional protein that causes the disease. And there's over 1,800 different diseases that evolves to some extent PTC mutation. We can think of cystic fibrosis, Duchenne, Hurler syndrome, there are many others, and also cancer, where in many cases, genes like TP53 can be inactivated through PTCs. Now, of course, in all of these genetic disease, there could be different types of mutations other than PTCs that can cause the mutation. For example, in cystic fibrosis, it is roughly 5% of the patients for which they have contracted the disease or they have the disease due to PTC mutation. So the compound acts by interacting with the NMD complex, and through this interaction enables the read-through, the translational read-through of the machinery across the stop codon, inserting an amino acid and then rescuing the translation of the rest of the protein, generating a merely or functional protein out of this in the presence of this compound. And the proof of concept of this was achieved in an in vivo model. And unfortunately for us, the multi-indication registration is needed in order to observe commercial viability. So the question to the science team is, what is the prevalence of other PTC-driven disease for which the actual mutation would be compatible with the MOA of the compound? And for this, we have divided the analysis into 2 categories, one focused on cancer, the other one focused on rare disease. Regarding the cancer, we have used complementary properties of 3 different platforms. One, C-Bioportal, that is found in the public domain, where we could identify tumor suppressor genes very, very quickly due to pre-calculated value data that can be associated to different type of cancer type. But the Genomics England platform was extremely useful for us for 2 reasons. First is that it is the exact same pipeline that is being used today in UK hospitals. Therefore, the mutations that are detected in Genomics England are the same mutations that will be detected by oncologists across the UK. And second, due to the study design, the cohort is reflective of the natural prevalence within the UK regarding PTC-driven tumor suppressor gene inactivation. And these results were confirmed in another platform called Project Genie, where there's more patients that have been prototyped per cancer type, but using gene panel technology, not whole genome sequencing. And the results were quite interesting. First, we did identify many cancer types where inactivated tumor suppressor genes due to PTCs were present and fairly prevalent. Another very interesting result is that there was quite some consistency in terms of prevalence estimation across these 3 resources, which was on our side quite reassuring. On the rare disease side, we used 2 approaches. One is the most, I think, the indication search was mining the tier 1 and 2 tables where the gene and disease and rare disease association is very well characterized. And within that group, to look on the number of cases where there are the inactivation, the causal variant that is inducing the disease is due to PTC. And we also use a hypothesis-free querying where we have looked for pair of genes that do carry PTCs and look if there's enrichment in a given disease or ICD-10 code or P code. And in both cases, we did find surprises or new associations that we did not expect. But in general, we identified a lot of PTCs, but they were very rarely the case that they were strongly enriched in one single disease, which can be expected. Therefore, a basket approach would be required for rare disease. So as a perspective is that GEL is also providing ways in order to rapidly test in a clinical trial or move forward using a clinical trial by identifying relevant patients either in cancer or rare disease, where they would have, through sequencing, they would have mutations that would be compatible to the MOA of this compound. And also another aspect that is very interesting is this network of stakeholders, which includes academics, patient association, and also industrial partners that could also join us in the development of this compound. The next example comes in diagnostic development kits. And it comes in the project team from Mayzent. Mayzent is, or siponimod, is a small molecule that is currently approved for the treatment of multiple sclerosis in Europe. And the team is currently proposing their FDA dossier for US market authorization. And the FDA is requesting that a companion diagnostic kit be settled for a genotype-based contraindication and dosing. And this is due to the fact that there is a rare variant on the CYP2C9 gene that induces a very fast metabolization of this compound, and in some cases will prevent efficacy of the compound. So we would like to either dose adjust or exclude patients based on that variant. So the team needs physical DNA samples in order to build and, more importantly, validate that companion diagnostic kit. So the classic approach is to do a population screen, and this is a very rare variant, meaning that we need to screen a lot of individuals, and we did a calculation that this would be between 25 and 36,000 individuals in order to have between 60 and 90 cases. Just in PCR kits, the estimated cost was roughly 2.5 million on average. And through our partnership with Genomics England, we were able to do a very simple query that took minutes. And we found 90 individuals that were carrying the desired or the required genotype. And through our collaboration, we secured access of these DNA samples, and now the team can develop a companion diagnostic kit. So the impact of this is that we accelerated our companion diagnostic kit production and also at lower cost. And there is also benefit for patients because they will have access more quickly to Mayzent on the US market and with higher expected efficacy and safety. The next example comes from Global Medical Affairs, and that is in oncology, and it is the capmatinib team. And capmatinib is a small molecule that is approved in several countries for adults with advanced metastatic lung cancer with MEK exon 14 skipping mutation. So this is a very rare indication. It's more has an orphan drug type of approval. But during the phase 1, the clinical trial team identified that solid tumors that are not lung cancer that also had variants that were not exon 14 skipping mutation showed signals of efficacy. Therefore, the key scientific question of that team was to determine and to catalog different types of MET mutation beyond these exon 14 skipping mutation, and in which cancer type. And the outcome was quite positive because we did find rare but new, that was not described before, of these MET mutation in a broad set of cancer types. Now, in terms of next steps, the collaboration with GEL enables us, through academic partnership, find the tumor blocks that of these sequence patients in order to perform IHC staining to verify if these new types of mutation do lead to hyperactive MET pathway. And if that is confirmed, using, for example, through GMS services, identify genotype, phenotype patients that could be considered for enrolling into a clinical trial, testing this hypothesis. Now, moving next to a more strategic level of discussion. So I hope I convinced you that Genomics England can provide a lot of value creation opportunities across R and across development. And actually, on our side, we realized relatively quickly that Novartis is not very well organized for such new type of resource that provides enterprise-wide return on investment. And when we think about it, this is among the very first resources where value is added from early discovery all the way to medical affairs, apart from basic stuff like Word or Excel or these type of resources. But in terms of scientific resource, this is really, really unique. But this also means that we need to disrupt how we're working internally. And one example from an organization point of view is that our budget structures are very fragmented, meaning that we need to rethink how we organize internally in order to make enterprise-wide investment decisions and also enterprise-wide investment accountability. From a data analytics and infrastructure, also, we always need to enhance our analytic capabilities, but also from a capacity perspective, this is also quite important because there is new needs that were not there before. From a partnership point of view, this is for us very key because a lot of these value creation comes from working very closely with external partners such as Genomics England. And what is very massively underappreciated is the importance of culture. Because, for example, the real world evidence teams never work with omics and never work with early discovery scientists who are very familiar with omics, but absolutely not familiar with real world data. Therefore, we're bringing together people that never work together. And also for our product teams and our discovery teams, awareness and also examples of what can be done can increase their culture and open our eyes to new opportunities. Regarding investment also into these partnerships, two key factors for us is really important is the quality of the data and also the relevancy of the data for the intended question. But what is really clear also is the importance of use cases within the company. So having a real cool and exciting data set does not suffice. We do need these use cases in order to ensure success. But despite these challenges, the reward are very interesting. So this is just a graphic of how we are translating value creation per medicine. And this is the revenue versus time. And of course, during R&D, all investments are liabilities until launch, which is in the middle section. But with working with such partners, we provided different ways that we can reduce R&D costs by reducing project attrition, by having higher quality assays, by identifying earlier biomarkers, and also examples of increased peak sales by finding novel markets that were unsuspected before. And all of this will provide a much favorable sigmoid curve. Partnership, of course, is really key. And partnering with GEL was extremely fruitful in many different aspects. And more as a provocative question, maybe for a discussion, is are there additional opportunities to partner across Discovery Forum members in order to further enhance these capabilities. So as a summary, so I hope I convinced you that GEL is way more than just a genetic database, that it has enterprise-wide applicability and the importance of partnership. And on this, I'll pass back the word to Kate.
Thank you, Pierre. First of all, let me welcome in thanking Pierre for a fantastic talk. Pierre, this was fantastic, and I think you've mentioned that at the end of your talk, that having a cool data set is not enough. You also have to have this evidence of use cases coming through, and I think this was spectacular coming from you from a perspective of generating new ideas for our partners, but also showing the participants who are listening that our data is really becoming used to make new drugs. And I think with that, I would like to open the floor to both within the room and on the chat to questions. There is already one question coming in from online audience, so I'll start off with that. And the question is, what level of transparency do patients have to how their data is being used? Can participants decide to what study partnership they consent for? So actually, maybe I'll start off with answering that question. That's probably more for us. At Genomics England, we have a very rigorous process in how we screen projects for being eligible to access to data. So in particular, companies have to go through a legal process, but also an ethical review process that is governed by Access Review Committee. And each project and each scientific question is evaluated on the merits of accessible uses of data to which participants have agreed their data to be used. And so all of the projects that are essentially being done in the research environment are reviewed, checked, and registered in that manner. So hopefully that answers the question from Anonymous. Is there any questions from the audience? I see there's one hand from Torben in the back. If you're posing a question, please introduce yourself and give your affiliation.
Hi, this is Torben. I'm from LifeBit, so we are behind much of the software of Genomics England. So number one question for us is obviously, Pierre, what do you think makes pharma users like yourselves, like Novartis and others that are in such new environment, what makes them successful? What do you think are the critical kind of requirements that you, from a pharma side, have for such trusted research environments?
Yes, so I think that accessibility is a very important one. For understanding accessibility, particularly to non-data scientists, in order to do easy feasibility-like question, to understand with my question, with my potential use case, is the data present very useful to answer it. And I think that what we found that this can be a barrier because data science is always a rare commodity. Therefore, if we can have tools that could enable more easy querying of aggregated results, that could be very useful to go forward. And I noticed that this was mentioned this morning, so I think that's a great step in the right direction for greater usage of the platform.
Fantastic. Any other questions from the audience? Chris? Yes, we need a mic.
Thanks. So Chris Wigley, Chief Exec at Genomics England. Pierre, good to see you, and thank you so much for such a wide-ranging and exciting talk. I might ask a double question if that's permissible. One is you asked about, you spoke really well, I think, about partnerships and about how we as an organization need to some extent to understand the complexities in your organization and kind of help you convene the right people. If you had a magic wand, what could we at Genomics England or the wider ecosystem in the UK do to make it easier to partner with us, access the the assets that you can get through Genomics England, but also sort of get access into the wider ecosystem. How can we help that whole rich family across Novartis kind of work together and kind of achieve your goals?
I'm looking for the wand. I think there's the difficulty in the question is that if you talk with it, you will get very different answer depending whom within Novartis you will talk to. And I think that, so if you have a research perspective, then having great multi-model data is of great interest, I think, to many, many scientists. So being smoother in or having more data generation opportunities, that would be great for even bringing this partnership to a higher level. From a human interaction point of view, I think it's fantastic. So I think don't change anything. That would be my recommendation. I think, for example, something that we don't find elsewhere is a great data science team that can bring new tools into an environment. And that is something that we appreciate very, very much. Also having maybe smoother interaction with the whole UK ecosystem, I think is also something that is very appealing. I think that translating findings that we have in a research environment into a prospective test, that is, whether it is in a lab through an academic collaboration, whether it is a clinical trial, whether it's through partnering with another partner in the Discovery Forum, for example, these are ways I think that would increase the value of this partnership in my mind.
So in a minute, you managed to conjure that magic wand. Hopefully this will give us some food for thought to work on improving our partnership throughout the next year, and bring you next year to raise the glass of London Pride with us next time we have a Discovery Forum. So with that, I'd like to close the session. Thank you again for being our keynote speaker for the day. Thank you for the audience for the participation, and I'd just also like to remind that we will be reconvening at 12:50 GMT time for the next session on genomic healthcare. So with that, I'd like to thank you again, close, and speak to you tomorrow, Pierre.